This is a production of Cornell University.
So today I'm gonna talk about, in a half fashion. The first of this half of this talk will be a summary of what we have been developing and deployed AI and robotics technology for breeding programs, specifically for grape disease resistant breeding. And then in the other kind of a half, I will talk about latest efforts just from our lab. Very kind of, if I count correctly, that's probably just the last month or so. But that make a kind of a total shift of from the tool space of using these technologies to more like an agent means it's more proactive and act as a human. So without further ado, I want kind of first to show all the people who are actually behind the work I'm gonna share today, not only myself, but across different labs with multiple PIs and also many of our fantastic students, postdocs, technicians who are actually doing all this kind of pioneering work to help us test these technologies and prove their effectiveness for plant genetics study and also to help facilitate the breeding cycles. Um, I use this, graph a lot for my talks because this is really my vision. In the kind of a past many decades, when human try to work with agriculture and food systems, usually being human directly work with the system, right? And many of us sitting here today will be more on the breeding side, which is the starting point of agriculture, in my opinion, right? We first need to domesticate some plant materials or maybe animal materials, and then we start the farming practices. And then that's a kind of a food chain all the way to the either the raw produces we can get and consume or into some of the formats that we're gonna produce food that can be delivered to our society. And in my vision, in addition to this traditional way that always being a human interacting with the system directly, I want to create a new layer powered by technology, so that the interaction is not just between human and plants and the nature, but also everything will be quantified and sensed by using this technology, so that we can have a more objective and quantitative understanding instead of just over years of the experiences. So as Nate mentioned, I got my PhD from Georgia, and I talked to several of the cotton growers, right? Because cotton is a big crop in Georgia, and some of them really mentioned, okay, I gotta translate my like a 30 years, experiences of how to grow cotton to my offsprings so that it can continue my success. But one of the challenge is even within the state of Georgia, climate change all the time, right? You start to have a lot of unexpected weather issues there. So the experiences may not necessarily always be successful in the future. So how we can use different technologies to empower that. And so back to kind of the whole supply chain, right? From breeding all the way to post-harvest quality assessment. And what we propose here is we can really have a multi-scale technologies that can measure or quantify some of the trees of interest across different scales. So like from molecular, many of you are working on genetics, genomics, right? And you provide the foundation, in my opinion, to the rest of the process. But then once we start to have this molecular level information, we form the organ, we form the individual plants, and all the way to a field or maybe forest, and then aggregated to a region and all the way to the whole global Earth system. And in my opinion, we should tackle all this information together rather than individually just, oh yeah, I'm gonna be the best farmer among my neighbors. Yes, you of course can do, but what if something suddenly happened, like for example, if there is a disease outbreak all surrounding you, would be fun? Probably not. So it's very important for us to take a systems approach to really look through the whole system rather than just saying, I want to improve that individual aspect. And today I'm gonna just talk about our actually 5 years efforts on plant disease resistant breeding and genetics study for grapes. It's a very well-known project called White Gene, right? And my colleague Bruce Raschke initiated that effort back to maybe 25 years ago. and we had the first kind of generation of the project. And then when I joined Cornell, it's approaching to the end of the Whites Gen 2 project. And then I joined the team for the Whites Gen 3, where we have a substantial shift from getting the basic genetics information, now try to use high throughput phenotyping to facilitate the whole breeding efforts and translation. And in my opinion, one of the statements when I kind of started my PhD dissertation is, oh, food security is a big issue, and we need to double the total food production, right, by 2050 to feed the rest of the world. I think this is a great, and ambitious goal. But then I talked with several of the plant pathologists, and they told me, all right, do you know, we actually lose about 15 to 30% of the total yield just because of the disease. And I was like, okay, we are trying to double that, but why a single aspect can cause up to 30% of the loss, right? So that sounds like a big gap that if we make efforts, we can help to bridge the kind of or solve the overall problem in our mind. And at the same time, many of the disease, problems must be still controlled using chemicals, right? And agrochemical is a kind of a significant issue right now. I actually visited Long Island maybe 3 weeks ago, and I noticed a biggest issue there is they have a pretty sandy soil down in Long Island, right? And then every single chemical they put into their agricultural production systems gonna easily dripping into the water table, and that's it's a kind of close to the largest city in the world, and people share the same water table for not only agricultural uses but also their drinking. Now that sounds like, oh yeah, there is a true human risk factor there, and we all don't want that happen. So that's the reason, we try the best effort to see whether or not we can come up with new plant materials, new technologies that can help us to manage this issue more effectively, that can significantly reduce the chemical applications while maintaining the needed quality and yield for the growers. So I felt like maybe 3 years ago, I showed that I only can show up to the middle part, which we have some field robotics and drone-based solutions for monitoring the disease progression. And now we really kind of close the full chain from the laboratory-based, Blackbird imaging robots to phytopatholobot, which is a field-based scouting robots that can do disease quantification. And we leverage the information from the ground and all the way tie back with remote sensing data from NASA and do a regional level prediction, and all the way to new hyperspectral systems that we are testing that can be integrated with harvesters that we can measure breaks or maybe other post-harvest quality, while also to detect whether or not there are any defects due to the disease pressure over the growing season. So that we really have the information throughout the chain. And we also have the new breeder, Maddie, who is kind of working on new projects that can improve the overall wine grape quality. So there are a lot of excitement to be shared. So I want to kind of acknowledge my colleague, Dr. Lance Tito Davidson, who really initiated this whole effort, proposed Blackbird, the initial version that can do microscopic imaging using RGB cameras. And then instead of for a human to sit in front a dissect microscope to check these pathogen growths, now we can use the robot actually to quickly image all of these cells and then tell you whether or not there are disease pressures. And then later on, we use these quantitative metrics to try to associate it with the genetic data to identify the loci that are relevant to the resistance, right? And then hopefully we can stack off many of these loci and translate into the breeding programs and yield a new plant varieties that can be naturally resistant to certain diseases that are in high pressure in our region or maybe across the country. But besides that, we actually receive a lot of interesting requests ranging from other crops, different leaves, disease, but also some of the insects, like in the kind of the lower corner here, you see that's actually some of the mice there, and they want to actually count the egg and also try to identify whether or not the mice may show certain behavior that gonna impact the past injury to the specific leaf discs they are interested in. So before that, again, they need to set all these things in a dissect microscope, but now we are able to just quantify them and use AI models to do that. So on the right side, that's actually our latest, effort. We call it a hyperbird. From the name, you probably can guess, it's a combination of the base robotic system from the Blackbird, and then we add the hyperspectral information for that. So it's a hyperspectral microscopic imaging robot. So for every around 3 hours, we can sample 384 leaf discs with up to 6 micron per pixel spatial resolution and zero point 0.5 nanometer per pixel in the spectral resolution. Okay, and then the good thing is we want to tackle to see whether or not we can push the time to detect any of these early signals showing the potential, infection. This is a collaboration with Dr. Kitty Goss' lab, and through some of the preliminary data there, we are showing we actually can push the day 24 hours after the inoculation. There are some special spectral signature already showing the infection in a very early stage, right? So many of you may be working on the breeding side. I think timing is very important. Now, in the past, we have to wait for 3 to 4 days to show the visible symptoms on the blackbird side. Now, with hyperbird, we can push for after 24 hours after inoculation. So every single step, if you can save 4 times of the time, accumulatively, you're gonna save a lot to translate this knowledge. And then I would see for agriculture, the biggest challenge is not the lab part, right? The lab part always being straightforward somehow, as long as we figure out how to intellectually, solve that challenge. But many times we need to solve the field performance because breeding is targeted to grab the new plant materials that can be really used for real world production systems. So how these lab findings can be translated into the field and what's their performance there? That's a kind of another question we want to tackle. And that's kind of related to high throughput plant phenotyping. I'm not sure if Michael Gore is around, but that's what when I kind of got inspired as a PhD student, they were trying to do some high throughput phenotyping work for cotton, and I happened to work with cotton, and just had a lot of mutual interest there. But now we're just trying to translate that to grapevines for the disease resistant study. And to me, having an ATV driven by human operator with the imaging system already being maybe a good solution, right, for many of you for phenotyping purposes. But I always kind of quote myself as a lazy engineer, which means if I can design something that can autonomously do the task for me, I would prefer in that way rather than I need to still go some manual efforts, right? So that's being coded as a lazy engineer, also being the motivation behind it. So if we want to turn that ATV-driven system into a fully autonomous system that can do the field phenotyping for us, we need to address two important components. One is navigation, right? So we just need the robot to just traverse, mimic what human scouter do, right? Just go one by one or plant by plant. And then the next step is a perception, is when the robot see this, grapevine, how you will be able to identify where the canopies are and where the disease symptoms are so that we can calculate the ratio. So if we have these two components and putting them together, right, then we will have this autonomous system. And here on the left, just showing a pure vision guided system. And on the right side, in the left panel, you see the bluish kind of line. That's actually a control signal to guide the robots. Just stick with that line, okay, in middle of that. So you won't hit any of the trellis system in the vineyard. And then the robot just navigates itself based on this information. And this is actually very similar to what Tesla is using for autonomous driving. And on the right side, you see the kind of a first version of this algorithm. We kind of develop a deep learning model that can segment where the canopies are in the image and then the disease infection part. And then we simply use the GPS to aggregate the information and then take the ratio to estimate the disease severity. So this is the first version of BPB. It should play automatically. Um, as I mentioned, while the robot is traversing autonomously in the vineyard rows, it sees the canopy through this specialized RGB camera, and you can see our model can do a near real-time, inference about where the canopies are and where these disease infections are, and later on to just aggregate all this information to provide quantitative disease severity metrics that can be used for downstream either genetic analysis or the breeding evaluation for the breeders to identify new canopial varietals that has a low disease pressure. So we've been testing this not only in New York, our research vineyards, but also deploy that in California, because many of the growers are very interested in how to using this mapping information for actual production management instead of just a breeding and research effort. So this is the latest version, and actually Cornell Chronic just shoot a new video, but this is Cole, actually a technician from Katie's lab. In the past, the Cole will be the lead to hire a group of 6, people that will be trained to identify the group disease. And now Cole is training or like a babysitting the robot, going through the vineyard and do the actual scouting work for himself. So for the research vineyard we are working on, for about 2.5 acres. In the past, for this group of 6 human scouters, they need several days to really do the disease scouting and turn that into some format people can use for analysis. And now for this robot, we can do every 2 hours to finish the entire vineyard and then do frequent updates for that part. And that's again another way. If the bottleneck is how fast we can evaluate these plant materials and their performance in the field condition. And that's where we want to substantially save the time for you all so that you get quick feedback and then make decision which direction you want to move forward in the next step. So we kind of deploy the robots much frequent than human scouter. And we found a very interesting thing is many of the times we talk about we need to do early or asymptomatic disease detection because that's going to be more meaningful for production management. And I would say based on our findings is as early as the second week, we deploy the robots. We already find, which is the second row here, identify the hotspot that later on being heavily severe, that's being severely infected by this disease. So that means not only about the importance of having asymptomatic disease detection, but also how fast or how early we can get early symptomatic disease detection so that we can inform people for decision making. But then that turns out into another interesting trace I propose back to the breeders and geneticists, whether or not there could be temporal responses from this genetic background that in the past we were not able to quantify them because of the constraint, right? Labor constraint. You can only deploy a group of 6 that many times or that frequent over a season to consider all this fatigue or like reduce the performance of a human operator. But now with these robots, we will be able to deploy them as frequent as we want, right? 2 hours, I don't think disease is gonna change that fast. So with this new information, whether or not we can quantify temporal traits, that's gonna change how the growers perceive their plant materials in the future. Like instead of always seeing asymptomatic disease identification for early or informed management, but more like a early symptomatic detection that can give them informed decision making already. So in addition to that, we also found the robot actually had improved scouting sensitivity, right? I think that might not be a surprise to many of you. When you kind of just get into the field and do your scouting, and you start to be very excited, yes, I want to look at my plant materials, identify those ones I either don't want or I wish I keep them forever, right? And then after maybe 15 minutes of a walk, it's like, it's a sunny day, but I might want to kind of do some other things, right? And then after half day of the scouting, you already decrease it to a point is, all right, I just want to finish this job. But then turn around, back to the robots. The robots only needs power. As long as we have a full voltage, battery supply the power to the robots, the robot is happy and working fine there. All right, so, and we found actually the robot identified the entire canopy and also identified those subtle infection regions that human might easily ignore. Because you're gonna just walk through your vineyard, it's not that kind of easy for you to keep concentrated on that small area for every single group line. You are very likely to miss something. But now for robots, you are able to retrieve that information. And we confirmed that for many of the parts, human scouter meets a certain, infected group lines, and the robots just pick them up back. Okay. So with this, we are very confident and say, all right, let's just deploy this system, right, for all the breeding programs or maybe genetic studies across the entire US, especially for the major grape producing states, including California, South Dakota, Minnesota, New York for sure, our home, and West Virginia. With different format, you can see still some of the locations prefer to have a human-driven ATV because of the infrastructure things that we can discuss later on. But for many of the places in New York, now we fully rely on the robots to do the scouting. But one of the challenge is how many robots we need, right? And I shared one robot now we can cover 2.5 acres of the grape vineyard within 2 hours, right, to finish the scanning. But what if I want to scan like 50 acres? And what if I would have multiple sites for testing my early kind of varietals, right? And how we will be able to handle the logistics of transporting all these robots and really deploy them for the needed kind of measurements there. So instead of just relying the robots, we think, okay, why not to leverage the remote sensing, right? As simple as we have a lot of drone-based technologies, but also NASA as a federal agency share a lot of publicly available data sets that we can leverage. But one of the kind of a bottleneck for remote sensing community is the oftentimes lack of the data for ground truths, right? They know what happened or what has happened from the satellite imagery. But what they don't know is what exactly happened on the ground. So how you can really just correlate the signal from this satellite imagery with something on the ground that are of our interest. So we propose instead of a human to do all this scouting, why not we identify those optimal locations and then use robots to do the scouting and supply the ground truth measurements so that we can enhance the data availability for remote sensing modeling. So we did two kind of pilot studies there. One is try to use a kind of a commercial available drone. What we did is for the same vineyard, we use the PPV to quickly scan that and get the ground truth. And then we just fly the drone right after that. And after that, we will just make a model. Now, instead of a human measurements or human ground scouting as the ground truth, we use the robot measured ones. And then we make a model, right, between the drone imagery and the robot predicted or robot scouted ratings for disease severity. And we reapply this model back to the same field and compare that accuracy with human scouting results, right? So we want to see how we can translate this knowledge and really just compare with the human performance. And results show that actually in the early stage, it's not that promising, right? Accuracy is about 60 to 70%, which means you still have a lot of errors there, considering this is a binary classification problem, right? Either infected or non-infected. But approaching to the end of the season, right, you start to see it's jump around 80%. And actually, we learned a very important thing is for this particular pastoral system, this is for grape downy mildew. Okay, it's not systematic. It's always a start with the lower part of the canopy and translate the disease symptoms or signals to the top of the canopy. Okay, it takes time. And remember, the satellite or remote sensing data is largely from the top view rather than from the side view. So much of the information in the early stage at the bottom of the canopy won't necessarily be reflected well on the top of the canopy. So this approach, at least from our kind of a study showing, pastoral system is critical about what's the sensing technologies you want to use to leverage the existing infrastructure. But still, if your goal is targeted for late season evaluation, more on the research side or breeding efforts, then that's still being a good tool. But if your goal is for early season or like a pre-breeding to quickly identify things you don't want, we probably need some alternative technologies in the future. And the other pilot study we tried is actually to tie back with the average scanning from NASA. It's an airborne-based high-resolution hyperspectral imaging system. It provides 0.5 meter per pixel resolution with this full spectrum coverage. And what we did again is now instead of only using human ground truth, which means a human say this is a pixel related to an infected group 1, or that is a pixel related to a healthy group 1, and then we can separate them, right, and do a basic statistical test to see what's the statistical power we can get to differentiate the two classes. We also use the robot scouting to do this kind of a differentiation and then do a statistical test there. And we showed actually an improved statistical power by actually using the robot scouting. It's a very marginal. Kitty and I are still looking into details about this, but at least numerically, it does show some advantages there. And more kind of evident in this case is for certain kind of a key spectral wavelength range, you would start to see better separation when you use the robot scouting as the ground truth to do the clustering and then try to do this comparison. So later on, again, we use the robots and human both as ground truths and train different models, and then reapply that to the rest of the field and try to compare what's the performance. And actually, we would see very comparable results. But again, for human, what you need is 2 acres, you need 6 people, and several days, finish one round. And now for the robot, you just need 2 hours. And you still need one person, and that one person is being required by regulatory, actually not by our interest, because we don't have any clear regulation now for how to using autonomous robots in the agriculture setup. But that's a kind of a scale difference, right? Multiple people, several days, versus now we just rely on a single robot and a couple of hours to scan the same field. And now if you use the same amount of effort and energy and resources, you can cover more. And I'm an engineer, correct me if I'm wrong, but in my opinion, breedings are usually being the number game, right? The more you testing or you kind of investigating, the higher the likelihood you're gonna find something that match with your desired traits, right? So it's important for us to improve the total throughput and capacity. And that's where the robots and AI can bring us. And this is another kind of a case study. We use the same system, and now from the grape downy mildew and powdery mildew, we actually tested this in California. This is a commercial vineyard with 5 acres. As I mentioned, usually being a week or so for human scouter. and rhubarb in hours. And now by using this closed loop, effort, by bringing rhubarb as a ground truth to train remote sensing model, it's just like in minutes. As soon as the NASA, finish their pre-processing of the collected data, we can apply the model and get all this disease infection information and just map them. And back to this slide, right? We said we already distribute the system across the entire US for major one grape breeding programs. And one of the interesting things from my eyes, like in terms of the data analysis, is how good my model can be generalized across different places. Because usually that's been the challenge for many of the AI researchers. Your model is not generalized very well. And we found our initial model does a pretty good job, but still have a little bit, glitches there when we translate from one state to another state. And with the latest, improvements in foundational models, and specifically in this case, large multimodal models, we saw substantial, improvements about model generalized capability. So for this particular model we developed, we do not only rely on the visual information from the images. We can just annotate and say, yeah, this is a symptom related to disease 1, that's a symptom related to disease 2. But we also use the text to regulate how the model needs to learn these features through the visual information. So basically, we just type in a text prompt. This is the image showing powdery mildew in the center, or maybe in the upper or left or right, or somewhere, so that the model will aggregate both the text description of the disease as well as the visual information from the image, and then do a joint inference to identify where the powdery mildew should be. And we test this model. Again, we use the New York data set we actually collected maybe 5 years ago and train the model, and then test it on New York, which looks pretty good. And then later on, we also test that in California data sets without any single tuning. So no California data set involved in this model training or fine tuning at all. And we directly got that into inference. And this is the result, showing a pretty good, visual appearance about where the canopies are, and the red actually showing the disease infections on these canopies. But the more quantitative, we actually did the test for both 2023 and 2024 growing seasons. The model without tuning achieved around 80% agreement with the human disease scouting. And we did not really find anything that goes beyond the 2 rating score differences for our models. So that means this model is pretty generalized across different situations. Like in New York, again, 5 years ago in New York, we collect the data, and then we train the model, apply to 2023 and 2024 data sets from California. The model performed almost as good as what we saw from the New York data set. And now with this kind of improved phenotyping accuracy, I think many of you are interested in that part. I'm also very wondering like how my improvements can really benefit the downstream tasks you are working on. And we did statistical analysis, and we found actually for both years of the data, by using the robot scouting, we can effectively showing the same QTLs being identified by using human scouts. But again, the working efficiency are totally different. Several people for a couple of days versus 2 hours for one people as a babysitter. Doesn't require any special training for that. Only needs to know some basic Linux commands. And also another thing we found is most of the time, the model kind of a scouting data gives us an earlier indication to identify the same QTL as compared to human scout. That means if a human scout in a later of the season to identify that QTL, we can now use the robot scouting data to identify the same QTL, but in a much earlier time. Again, although grape is a little bit different because it's a perennial crop, it's a little bit hard to pull it out from the ground. But let's imagine if this is a annual crop. That means you can see another month And sometimes that means you could have a 2 rounds of field tests in the same growing season for testing your different candidates. And I think that's going to be a substantial, advantage for many of the translation work from genetics understanding to the actual breeding effort. And also, what kind of amazes us is in the past, we have these scores, and then they just compare for a single year and do analysis and then conclude that, right? But now we have this kind of robot data. We can show not only the same year, but really compare the image differences over the years. And we found several of these kind of special cases that either we have a stack of these genes supposed to be controlling the resistance to certain disease and really showing well kind of effects, but some of them being already break out by certain things. And again, in the past, we are mostly focusing on what's the plant material's genetic background that can give us the needed resistance or advantages. But now with these improved efforts, we identify new opportunity that we can include additional dimension. And in this case, will be the pathogen mutants, right? Not only the plants is involved with the human desire and efforts, but this pathogen naturally evolved as well. And sometimes they can just evolve to break down what we bring into this system. And now with the robot kind of and AI help, we probably can facilitate that speed and also start to consider that dimension and hopefully to tackle this problem in a more holistic approach. And the last piece, as I mentioned, we have some berry defect detection, just a regular hyperspectral imaging system. And we have been testing this method for the past 3 years, very stable, achieve pretty good accuracy in identifying either the berry moth damage or powdery mildew on the berries so that we can introduce into the post-harvesting session and also can be connected with the berry breeding programs. And up to this point, you already identify, all right, so is this a research, right? I think that's the exciting part, but also a little bit boring during the transitioning period. We now have the capacity, we have the knowledge, we have the collaborators, we have these interdisciplinary teams, and we can repeat this process many, many times and drive us to the success. But all the way when you kind of start a new project, although the final goal, the motivation is different and excites you, but the paradigm is almost the same. We have some observations, we start to have our hypotheses, and then we do or design experiments and conduct these experiments, gather data, analyze them, interpret, and publish. And hopefully, we start to have some new observations and crank the engine forever. That has been, in the many decades in the past. And I think this picture gonna be very familiar to many of you, right? Gregor Mendel, which is the father of modern genetics. But also, I want to point out Alexander Zawadzki, who somehow served as his unofficial mentor and always encouraged him, right, to explore things. So for me, the kind of a part of our research paradigm is exciting because of 2 aspects. One is these human aspects. We always get encouragement from our colleagues, from our mentors, so that we keep exploring things that we are curious about. But also the other part is, yes, we need to do this actual legwork. And that's where, again, a lazy engineer wants to automate that part so that I don't need to do the actual work. So there are a lot of kind of ongoing efforts for AI in science, especially for chemical engineering, because many of these kind of experiments can be controlled in their facility, right? The reactors, and it already being kind of modernized with robotic control. But for many of the plant science, studies, especially involving wet labs experiments or field experiments, this is still being the empty space. And we try to kind of fill in that. And we propose Alex, which actually I got inspired by the story between Alexander and Greg Mendel. And I want to create an AI system that can encourage human researchers, right? So that we keep our excitement, but also help us to automate many of these processes. So basically, this system in the current stage, we have the domain scientists, and we have our data analyst, and we have the machine learning engineer that can implement some of these methods. And then we feed in with the literature human selected for the domain scientist. And for the data analyst, we just relying on the pre-training knowledge for the general data science. And then for the MLE, we send to this agent a couple of documents related to APIs for specific libraries that can handle classification and regression automatically. And then we kind of want to test this new AI system, and we see why not to test the plant sampling strategy, right? Because this is a grape red blotch disease. And one of the things for the remote sensing kind of a study is you need to ground truth, right? But instead of a research vineyard, they being sent to the commercial vineyards in Napa Valley, and that's the picture. All right, so that's the millions of these grapevines. And as a human researcher, so you need to either exhaustively do every single one, virus testing to confirm that's a red blotch, or you're going to need to come up with a sampling strategy, identify the plant tissues, and that can inform you where are the pixels I need to use for training my remote sensing model. Right, so it's a very tedious work, and the human usually based on experiences and hard to control its quality. So we just pull the data from the historical records and then challenge our AI agent system. And here are some kind of interesting observations. This is the first kind of iteration. What's the inputs are? First, we have selected the literature, okay, for the domain scientist agent. We have a historical data from 2021 to 2024 about the grape red blotch disease infection in that vineyard. And we ask the question to this agent system. We want to figure out how to build a prediction model to identify the grape red blotch disease. That's it. That's all what we need to do. And the rest is being handled automatically by this system itself. And the first thing is we did not identify which column in that CSV file is the predictable variable. The agent can identify itself. And somehow, after kind of glancing at the first several rows of the data, the model decided, okay, I'm going to go for regression for this problem instead of classification. Again, no human guidance, fully autonomous. And then going just forward for a kind of iteration 18, I start to see some promising trend of using classification. I will explain it just jumping back and forth and see, yeah, this looks very promising, but also share some critics that we lack certain features we want to use to improve the model performance. And then this performance being realized in the next iteration, again, automatically, and yielded the best model. So we did this kind of a problem testing and quantify their performance multiple times and try to just see what's the performance there, right? So we roughly counted for a human group to tackle this challenge, usually at least it takes several weeks of the effort. And now with this system, it's just a couple of hours. We finished the whole round of the new iteration of the research discovery. And we yield a model that gives us a pretty good, sometimes a very decent, performance for this problem. And also, this system does not only end up with, oh yeah, that's a model you can use, but also generate the papers and even draw the figures that we can share. And we just, submitted this paper to a conference called Agent for Science that AI is the primary author for this paper, and AI will also serve as the primary reviewer. So I haven't got any response yet, but I will share back with the community. So I gotta just wrap up here, right? Because AI as tools, I think already show very proven track record to improve the accuracy and throughputs that can help, the understanding of genetics and the breeding programs. But I think the next step is really AI as agents that can achieve full autonomy and help us to fill in a lot of gaps into the current scientific discovery processes and really make a paradigm shift from just as tools, but as now our partners. And I will keep sharing more of our updates, but in the past several weeks, we keep doing new kind of case study and exploring new things. And I'm very open and willing to work with the SIPS community and to identify new case studies that we can challenge this system. So with all that, I just conclude my seminar, and hopefully I still have a couple of minutes for questions. Yeah.
Hi, I want to know how your robotics recognizes the affected plant. I'm not in this field, but curious about it. And plus, single grapes should be affected by several diseases. So kind of robotics recognize different disease. Plus, if these robots function well on the good weather, it can still work well at bad weather.
These are great questions. So basically, three questions there. Number one, how the AI model really identify the disease infections in the images. And basically, so we use a supervised machine learning training, right? And human just on the images, just label where are the regions showing the disease symptoms. And then we're gonna use that as a ground truth to guide the model training. So later on, the model will learn the features to just generate the final disease mask will match with the human annotation, right? So that's the way how the AI models can gradually learn the features and then identify the disease in the images. And for our next question, there could be multiple diseases, right, in a single vineyard, and how the model will be able to deal with that. That's excellent question. So far, for many of the genetics studies and breeding programs, we usually kind of select a particular trait we want to improve at the moment, especially for the current project I'm involved with. And that means for powdery mildew, which has a very different appearance than grape downy mildew, we can either just put them individually or we can combine them together. But there are many kind of disease symptoms share the kind of appearance, which we are still trying different ways to figure out how to differentiate them more effectively. And one of the ongoing efforts is we develop a new generation of the phytopathology bots, include both thermal and hyperspectral imaging. And we hope those signatures can help us to differentiate this disease together, okay? And the last question is about the weather, right? So the robot itself can run in raining days, but our question is actually, is that meaningful or is that informative for people to get the information during the weather that is not suitable for the robots to do anything? Or maybe there is, it's not a kind of a straightforward answer. I would say we probably can just do some tests to see what's the quality of the image we can gather during a raining day or snowing day, and then try to see if we can identify some new trees. But I think in general, our kind of experience is once you have new capacity, you always observe something new that in the past, either because of different constraints, especially resource constraints, you cannot do, or now you just, wow, this is interesting. I want to further look into that. So I think, again, that's the new opportunities by using these technologies can bring back to human researchers.
This has been a production of Cornell University, on the web at cornell.edu.